Gel electrophoresis is a way of separating molecules based on their size. So DNA based on how many base pairs, RNA based on how many nucleotides, proteins based on how many amino acids. And the key is a gel here that you melt, place into a mold, let it set. And when you look at it in the end view here, it has all of these tiny little pores. And it's going to be those pores through which our molecules run. And I have, so what size fragments will get through those gel faster? And the important point is that the smaller the fragments are, the faster they will run through the gel. They'll be able to get through those pores more quickly than the larger fragments. And that's how you're going to separate these fragments of DNA, based on how fast they migrate through that gel. Now, there's two types of gels that you can use. Acrylamide resolves small fragments best, and agarose resolves large fragments best. And then on top of that, you can adjust the percentage of acrylamide or agarose within a gel to even then more adjust the resolution of that gel. So the basic way it works, let's look at that running DNA and RNA, is that you've got the gel, you have put a buffer, right, that conducts electricity, over top of the gel. You then have a negative electrode and a positive electrode, so you're going to run a current through that gel. You incubate, you dispense your DNA into the well at the top here, and then you run the current. Now, DNA and RNA are both going to run towards the positive pole. Why is that? Let's think about it. DNA, RNA, they both got those phosphodiester bonds. That phosphate group has lost a hydrogen, so it's negatively charged. So all DNA, all RNA is negatively charged. And so we know all of that will run towards the positive pole. Now, RNA differs in DNA in one respect that we have to pay attention to, in that RNA is single-stranded, and so it forms these complex folded structures. They can be very compact. The problem with that is that RNA would tend to run through that gel not based on its size, but based on how compact it is, and we don't want that. So, for RNA gels, you do all the same things that we show here, but on top of that, you have to add to the gel itself. You have to add to the RNA sample itself formaldehyde, and formaldehyde will keep that RNA denatured from folding up on itself. So in that way, even the RNA is going to run through that gel based on its size and not how compact it is. So after a certain amount of time, the longer fragments are going to migrate slowly, so they're over here. And the small fragments are going to migrate very quickly, so they end up down here. So you can take that gel out, and you can stain that gel with various stains that then allow you to visualize on a gel where the various fragments of DNA are. So let's go through some terminology here. So what we have coming down from each one of the wells, we call these lanes. So at the top are wells, that's where you loaded your sample, and then extending down, we call that a lane. So lane 1, 2, 3, 4, 5. And then within the lanes, we see what are called bands. A band represents a bunch of DNA or RNA molecules that are all of the same size. Now, the bands that are lower on the gel, they're going to represent smaller fragments of DNA. Bands that are higher up on the gel are going to represent larger fragments of DNA. Now, proteins are different than DNA and RNA in two ways. The first is that same problem that we had with RNA, is that proteins form complex structures. They fold in on themselves. So we have to have a way to denature those proteins so they run through the gel based on their size and not based on how compact they are. In addition, proteins, unlike DNA, unlike RNA, proteins aren't necessarily negatively charged. Some proteins might be. Some proteins might be positively charged. Some proteins might have a neutral charge. So we have to have a way where all proteins have the same net charge. And the way we do that is incubating those proteins with a molecule called SDS, sodium dodecyl sulfate. And SDS, as you can see, is negatively charged. It binds to these proteins, gives all of those proteins a negative charge. On top of that, it helps denature those proteins, along with heating it and adding mercaptan ethanol to break the disulfide bonds. So in this way, incubating proteins with SDS and denaturing them, now you can run them through a gel. They're going to run towards the positive pole, and they're going to run based on their size and not how compact they are. Okay. But then after that, it's going to be the same thing. You take the gel out, you stain the gel, and then you see bands appearing. And bands represent proteins of the same size. The farther down the gel they've run, the smaller the protein. The less they've run through that gel, the larger the protein. Here is a nice table that shows you what type of gels you can run these through. Notice protein you can only run through acrylamide. Proteins are too small to be resolved well in agarose gels. Do you have to denature it? Yep. RNA you've got to denature with formaldehyde. Proteins with SDS. DNA, nope. And then what kind of stains you can use to visualize. Now, nowadays, actually, this isn't so much true. Ethidium bromide for staining DNA and RNA isn't used as much because it's a mutagen. Instead, there are some non-toxic stains that can be used to stain RNA and DNA. And so you might get gels that look like this. So here you might get something that is not so complex, where you can see individual bands. You can see individual bands. You can see individual bands. But sometimes if the source of DNA that you used is super complex and has a ton of different fragments, then what you'll end up with is what's called a smear, where you can't detect really any individual band. We'll talk about how you can find individual sequences of DNA within this gel in our next talk.